Dear students, hope all of you are staying at home and doing fine. In this session, let me discuss with you the application of bacteriophages in nanobiotechnology. I do have sent all the materials for Unit 4 and 5 through your Google Classroom, and in continuation with this, I'm trying to explain each topic to you through this forum. In this session, we'll be concentrating the applications of bacteriophages in nanobiotechnology. So, as you all know, bacteriophages are viruses that infect only bacterial cells. These are the viruses. Now, we say viruses as biological entities, but without a host cell, they are non living entities. They are obligate intracellular parasites. Now, what does obligate intracellular parasites means? So they require a bacterium. Obligate. Obligate means strict. So they are strictly intracellular parasites. They need a host for their multiplication. So what do? The common structure of a bacteriophage is that they have an outer capsid and they have a genetic material which is packed inside the capsid. Once they enter into the bacterium, they utilize the machinery, that is, the enzymes, the proteins which are involved in the bacterial DNA replication. So they either integrate their genetic material into the bacterium and remain as prophage or lyse the bacteria, and thus they are going to utilize the protein and all the other machinery which is available in the bacteria for their own replication, and thus they sustain or live inside a bacterial cell. That is why they are known as obligate intracellular parasites. Now, based upon the genetic material which they harbor, that is, what type of genetic material is found inside the capsid, within the capsid, we classify them into four major groups. That is, DNA phages, those which are single stranded DNA phages, double stranded DNA phages, single stranded RNA phages, and double stranded RNA phages. Now, let us forget about these last two, that is, the single-stranded RNA phages and the double-stranded RNA phages. Now we have dealt with in detail about the single-stranded DNA phages. So the classical example being M13 and the classical example of a double-stranded phages being lambda. So we have dealt with these in detail already. In our previous classes, like genetic engineering, we will deal with this, the genetics and how they are utilized as cloning vectors. Though that is not our interest right now. We are going to see how these phages are manipulated in nanobiotechnology and how they are used. And this is a common structure that we have dealt with it in a large, like we have discussed with it for a long time. You know this, right? So how the bacteriophage lambda is packaged, or how, what is the basic structure. So this is a schematic representation of a lambda phage. So the lambda phages, you know, now these are the classical structures, like you have a head, okay, you have a contractile sheath, and these are the tail fibers. So the head harbors the genetic material. So here the genetic material is double stranded DNA. Double stranded DNA is the genetic material. And this is the structure of a filamentous phage. So that is your M13 phage. Now coming back to this again, you have the bacteriophage lambda, which has harboring a double-stranded DNA within. It is now protected well by a capsid coat. And now what happens? The basic steps, like these are the tail fibers which are used to adsorb to a host. Now this, if this is your E. coli, now these are the tail fibers which adsorb onto the bacterial host, and then the lambda phage sends in the DNA within the host. So these tail fibers are utilized for binding of the bacteriophage or adsorption of the bacteriophage on the bacteria. Is that clear? So the double-stranded DNA then is injected into the host. So it takes up the host machinery. Now let's see the life cycle of the lambda phages. So as you can see, so there are two types of life cycle. One is the lytic cycle. So this is the lytic cycle, and this is the lysogenic cycle. So in the lytic cycle, as you can see, the lambda phage enters into the cell, injects the bacterial genome, and then it is going to make the bacteria synthesize the bacterial genome and also the capsid proteins, and they will go for a process called as packaging. That is, each coat protein is packaged within the lambda head and the tail assembles. So as you can see, it is assembled, and then final step, it lyses this bacteria, and then it is going to get released. That is, it kills the bacteria finally. But in lysogenic cycle, it remains as a prophage. So a prophage means a bacteria which harbors a phage genome. So the phage genome enters, and then it is integrating, as you can see. So it integrates and remains as a prophage. And now this is the structure of a filamentous phage. So the filamentous phage, the classical example here we are going to talk about is your M13. So they have a outer capsid and then a single stranded DNA which is packed inside, and you can see these are the group of proteins. So the group of proteins, so we have these proteins, the name of the proteins and their characteristic features are given here. So you have the protein 3, 6, 8, 7, 9, okay, the 5 proteins which are present at the end of the tail fibers of the filamentous phages. Now we are going to see the application of the bacteriophages in nanotechnology or the nanobiotechnology. We manipulate the bacteriophage. We have seen earlier how we manipulate the bacteriophage for use as a cloning vector. Remember, like we have spliced the non-essential genes, that is those genes which are not needed for the lysogenic cycle, right? So those genes, because we don't want a lysogenic cycle when it is used as a cloning vector, we cut those genes and we insert a foreign DNA and use them as a vector. So those apart, keeping those apart. Now similarly, you can manipulate a bacteriophage and use it as a for your nanomedicine. Now the first and foremost technique that we are going to discuss is your bacteriophage display. Now bacteriophage display. So the bacteriophage particles, as you know, they exhibit a high level of organization. So they are having a highly organized genetics, okay, genes, and they follow a pattern of replication and the assembly. The major role that bacteriophage or the major characteristic feature of a bacteriophage which is useful for nanobiotechnology is a self-assembly, right? So they assemble themselves. The bacteriophage, we have seen this already. I'm sorry for this. As you can see, it follows a pattern. It assembles, the self-assembly. So these proteins, they are synthesized, they assemble themselves. So it follows the organized pattern. So this self-assembly is a characteristic feature which is applied in your nanobiotechnology. The first and foremost application being bacteriophage display. So both lambda phages and the filamentous phages are utilized for this purpose. But the most easily manipulated one is your filamentous bacteriophages. So the use of bacteriophage here is that We are going to make the bacteriophage particles to display exogenous peptides, right? So what does that mean? We'll just go into that. Now the applications being epitope identification. The bacteriophages display the uses being epitope identification. So epitope means the region on an antigen which is determining the antigenicity. So that is what? The sequence which makes the particular compound an antigen. So it is used for epitope identification, antigen delivery, drug discovery, vaccine design, and targeting of eukaryotic cells, bioimaging, biosensing, enzyme display or design of materials. So these are the various applications of the bacteriophage display. Now, what is this? Very simple. Bacteriophage particles display exogenous particles. How does it do it? So, a first method that we are going to discuss is your molecular manipulation. Now, what is molecular manipulation? The exogenous gene, that is, gene of your interest or your foreign gene, right? It is now spliced. Spliced means cut, and it is now ligated along with the bacteriophage genome. So, your exogenous gene is fused with your bacteriophage genome. So, this is a fusion protein. Okay, so this fusion protein is now found on the bacteriophage surface. Is that clear? So you have a so this is the exogenous gene. If this is your bacteriophage gene, now this is integrated into this, and now the bacteriophage will display this protein on its surface. Okay, so literally it is known as a surface decorated. Is that clear? So the surface decorated with the product of expression of the foreign gene. So this is what we mean by a bacteriophage display by molecular manipulation. So what is the application of this molecular manipulation? So when you are doing a bacteriophage display, you can immobilize an antigen moiety of interest on a surface that has been exposed to an engineered bacteriophage library. That is, the first step will be your construction of bacteriophage library, right? So this bacteriophage library will be displaying the protein of your interest. Now those bacteriophage particles presenting molecules with affinity to the immobilized antigen will therefore bind, and the rest of the library will be washed away. The bound bacteriophage fraction can then be eluted, amplified, and resulting in a concentrated mixture of bacteriophage particles presenting the relevant particle. So this procedure, which is repeated again and again, we call it as biopanning. Now this is the biopanning technique, the schematic representation of the biopanning technique. So let us start with this cycle. So this is the phage library. Phage library means there are a number of or a collection of bacteriophages which display, as I told you already, which display the foreign protein on their surface. Now this phage library, from this phage library, you are isolating or purifying the phages, the second step. Now after this, there are bacteria, okay, which is or any immobilized, okay. So the antigen is immobilized on a surface. So you're treating this immobilized surface with this phages. So those which are having affinity will bind to the surface, and those which do not bind are washed away. Now those which are bound are selected or purified again, and then the cycle goes on. They are propagated, and this cycle keeps on going. And this is the technique of biopanning. Is that clear? Now, there is one more approach to the bacteriophage display apart from molecular manipulation. So, this here involves chemical modification. Instead of your molecular manipulation, here we are using chemical modification. So what is chemical modification? You are attaching the molecule of your interest into your bacteriophage particle. So you see the bacteriophage is decorated both inside and outside. Okay, so here it does not need any manipulation with the DNA. So what happens is it is a safe method because the genetically modification organisms, the genetically modified organisms, we call it as GMO. So they, if they are going to escape into the normal mechanism, normal, so what happens, sometimes they might lead to the, they might infect the wild phages and they might revert the wild phages also. So some sort of mutation might occur. So it might not always go in the right direction if not done properly. So to be safe and from the practical point of view, this is a very important advantage of a phage display technique, that is the chemical modification. So here it does not involve the DNA, it is purely addition of a chemical to the bacteriophage surface. Now the third approach is your competitive phage display. So this method allows for presentation of foreign peptides or proteins on the phage surface, again without any modifications in the phage genome, right? You are not changing any of that of the phage genome. You are allowing the peptide to be just expressed. Since protein fusions are expressed by a engineered bacterial host, that is, you're already having a bacterial cell, a host, which is harboring a expression vector, and this synthesizes a fusion protein. I repeat, you have a host cell, that is E. coli, which is having a expression vector. We know what are expression vectors. These are the vectors which are designed for maximal expression of your protein, right? They do have a promoter, they do have a RBS, they have a termination sequence which is recognized by the host. So this has an enhanced expression of your foreign protein. Fine. So you have a bacterial host which harbors the expression vector and synthesizes a protein. Fine. So this host is now utilized for your bacteriophage display. That is, now we are infecting this host again with the phage which carries the surface. That is, on the surface it carries your foreign protein. So now what happens? In the same bacterial cells, when infected by a target phage, wild type phage proteins are also produced. So both wild type and the non, your targeted protein carrying phage, both of them are allowed for propagation in the bacterial cell, right? Now there is a competition between these two. Wild type and fusion proteins are randomly incorporated into the phage capsule. So that is why it is called as competitive phage display. So there is a competition between the wild type and fusion proteins for their integration into the phage capsule. So this leads to a competitive phage display. Again, from the GMO point of view, that is, this is again a safe method than the first method. So now till now we have discussed three methods. One is molecular manipulation, two your chemical modification, and the third one competitive phage display. Now, bacteriophages can also be used as a vaccine carriers. Fine. So they are harboring a capsule. Okay. They are having a capsule and harbors a genetic material. Fine. So the protein coat was utilized for bacteriophage display or the phage display technique and biopanning technique. Here we are going to utilize the other features of the bacteriophage so they can act as vaccines. Now there are two approaches for this. One is the phage display vaccine, the other is the phage DNA vaccines. Now in phage display vaccines, what is the procedure? So here they aim at producing immunogenic bacteriophage particles. Now here again the surfaces are decorated with the foreign antigens. In the bacteriophage display, what we did, we made a change to the DNA such that it is going to express the foreign protein on the surface. Here the antigen is expressed as a fusion product and on the one of the structural protein is going to be a foreign antigen. Okay, so phage display vaccine, similar to the bacteriophage display technique. Another one is your phage DNA vaccines. So DNA vaccines, as the name suggests, how they are produced, you incorporate the foreign antigen genes in the bacteriophage genome under the strong eukaryotic promoters. So the bacteriophage is a manipulated one here. It is genetically modified. The protein is not changed anyway. The DNA is now combined, ligated with the foreign antigen. Whereas in the previous case, the changes is made on the structural protein. In the next case, that is the DNA vaccines, there is a change in the DNA. So these are the two methods. So the two distinct ways is one, by direct vaccination with bacteriophage particles carrying vaccine antigens decorating the surface. The second methodology is by using bacteriophage particles to deliver the DNA vaccine, okay, which is an expression cassette. Cassette means a fusion, right? A fusion of those two different genes incorporated into the bacteriophages. So to summarize, there are two methods. One is a phage display vaccines and the other is the phage DNA vaccines. The third mechanism, how bacteriophages are used as for gene delivery. So the well-known mechanism of the DNA packaging within the viral capsid, it is utilized here. So they can act as literally as nanomotors. Fine. So this particular work was done by Harry et al. In 2014. So here the bacteriophage 529. Now, this this bacteriophage is a double-stranded DNA phage. Okay. Now, this phage, or it utilizes, we are going to utilize the feature of this bacteriophage. Three proteins, that is group 10, the connected protein, group 16, and a small packaging RNA. All of these three are needed, okay, along with the ATP. They will push the DNA into the procapsid, and now this mechanism is utilized. So you are literally using this as a nano cage. Fine. So the RNA is, okay, the bacteriophage RNA is now within, now this capsid. In this capsid, you are going to put in the DNA of your interest, and now you are going to utilize the machinery of the bacteriophage to push the DNA of your interest into the host cell. Fine. So they are used as a nano cage. The nano cage means, now how we know what is a cage is. Now within a cage you can put in anything. You can like lock anything, or can something can be sealed. Similarly, in the bacteriophage capsid here seals the DNA of your interest. And now when it is going to infect a host, the machinery of the bacteriophage, that is the three major components, group 10, group 16, and your RNA, will push your DNA of interest into the host. So this is actually utilized as a carrier material. Your capsid now, or the bacteriophage as a whole, is going to carry your DNA of interest into your host. Fine. And among this, when you compare, M13 is more preferred than the lambda phages because of the tendency to migrate toward the blood vessel walls when administered in vivo. Okay, so they go and bind to or interact with the receptors in vivo better than your lambda phages, right? So that is what, to summarize, bacteriophage particles can thus be used as designed nanocarriers for the targeted delivery of both therapeutic agents and genetic sequences. Okay, so what is the major role of phage coat here is to protect the DNA from degradation after injection, and the ability to display the foreign molecules on the phage coat, enabling the targeting of specific cell types. Now, apart from their use as a gene delivery carriers and also vaccine carriers and display proteins, the bacteriophages can also be utilized as a biosensors. So here you are chemically attaching any groups. Okay, so this can be utilized for biodetection system. So bacteriophages can be used for biodetection. Now the classical example which is discussed here is the biotinylation of T4 bacteriophage. You engineer the T4 bacteriophage to express the biotin binding domain on a capsid. That is, you have the T4 bacteriophages. T4 again is a bacteriophage which infects E. coli, right? So this phage will be expressing biotin binding domain, okay, on the capsid, that is on the exterior surface. Okay, so this again you develop by bacteriophage display technique. Now they can bind to a streptavidin, always binding of a biotin and streptavidin. We know biotin always binds with the avidin with a higher affinity. So you have a gold surface which is derivatized with streptavidin or streptavidin coated magnetic beads, or one more microcrystalline cellulose beads. Okay, three of this, any one of this, okay, any one of these three surfaces has been reported. So this T4 will carry the biotin binding domain on their surface, and you have another surface, okay, which for the receptor, these are ligand. The T4 is a ligand. You have a receptor. The receptor might be a derivatized gold surface with streptavidin, or derivatized with streptavidin magnetic beads and microcrystalline cellulose beads. So this can be used for a biodetection system. Now to continue with bacteriophage based nanomaterials. Apart from the organic materials being used, we can also use inorganic materials. Fine. Inorganic materials. In this case, let us see, you are using zinc sulfide and cadmium sulfide. So they can be used as nanowires and nano rings. Fine. So in this case, the high density of nucleating peptides along with the filament of the filamentous phages, right? So they are used to create inorganic nanostructures, single crystal nanotubes. So here the nucleopeptides are fused to P8. Remember, the P8 is a protein of M13. Okay, so now this is expressed from a phage. Now what is a phage? Phage, remember, phage are hybrid vectors. They are constructed with fusion of M13 and then your bacteria like plasmids, bacterial plasmids. So plasmids and phage vectors fuse together. The hybrid makes up a phage. So the phage here expresses the P8. Fine. So this is assembled along with the nucleating peptides. Now this is exposed to zinc sulfide solution at 0. Now what happens? This results in the formation of a tube of nanocrystals, right? So this is a nanowire, right? And this is a semi-conducting material, right? Similarly, nano rings. Now here, this nano rings can be produced by adding different functionalities to the ends of the M13. So at the two ends of the M13, here there are two binding functions. One with the nickel molecule, other with the avidin. Fine. So if this is the phage, it is bound here. Your nickel molecule, avidin molecule is now bound here. Fine. Now was used in conjunction. So these two are used in conjunction with the phage that carry ligands for binding specificity. So these act as a ligands. Now you have a polyhistidine peptide that binds to the nickel ions, was displayed at the P7 or P9 end of the phage, and the streptavidin binding peptide was displayed at the opposite. So here you have the binding site for this nickel. Here you have the binding site for avidin. Okay. So when you have the adapter, this adapter molecule, when it is mixed with this displaying particle. So you have the M13 which displays the binding site. You have an adapter molecule which has these two. So they bind and they make up a nano ring structure. So this structure is a nano ring structure. And to continue, I'm just talking about the phage method. So we talked about how phage method is used for synthesis of a nano ring. Now, as I explained you already, these are a class of vectors which carry plasmid and the phage origins M13 F1 origin of replication, and they have used for phage display technology. Fine. So this is just for your convenience that I have explained over here. Now let me summarize with the application of your bacteriophage as the now let me summarize the application of the bacteriophages in nanotechnology.